Hello Biotechnicals. Welcome back once again to Biotechnica's YouTube channel. Today we'll look what are some of the few reasons why you get Part C questions wrong in CSIR NET exam. I'm sure many of you would have already faced this situation. Maximum, we understand Part C questions have 4 marks of maximum questions. If you get it correctly, you get the marks. If you get it incorrect, -1 is deducted. Not much, but that -1 can make a difference when it comes to the overall marks. And because we are tempted to get the marks, we tend to overlook some of the details. We don't think properly, and that's why we get most of our questions wrong in Part C. After the exam, you will be very happy that okay, I have solved many Part C questions, I should be clearing the exam. But not only you lose the 4 marks upon getting them wrong, but you also lose additional negative marking. I'll tell you some reasons. If possible, please rectify those on your own. First reason is taking a common approach to solve all types of Part C questions. There are many types of questions which come in Part C. The most popular ones are true or false type of questions. Another one is graph-based type of questions. Match the following. Arranging or ordering something. The sequence of some pathway or some technical process, or arranging on the basis of some characteristic. Experimental analysis, where you will be given an analysis data, an experimental data, and you have to draw conclusion out of it. Diagram based, either it can be process, pathway, signaling pathway. They can ask what type of, they can mark some things in the diagram. For example, if you check this purine ring over here, they will give you the purine ring and they will ask you which is what. Like N1 is what? It's from aspartic acid or it's from glutamine. N3 is what? It's from aspartic acid or glutamine. C4, C5 is from what? So such kind of diagrammatic questions can be asked, or they can give a signaling pathway that this particular, let's say the WNT signaling pathway. So it is giving a signal to some other kind of molecule on the receptor. So what kind of receptor is that? They will mark it B. What it is triggering, they will mark it C. So what is A, B, C? They will ask you. This kind of diagrammatic questions can also be another type. And another type is numerical type of questions. Now I hope you understand that there are different approaches for all this kind of questions. Like for numerical problems, for example, you have to first write down what are the given things, what is already provided. Then you have to apply the correct formula, the appropriate formula you have to figure out based on what are the given fact. You have to do the unit conversions. For applying in a formula, you cannot use different units for several terms, okay? Then you have to calculate properly without making any calculation mistake. And then you have to convert the answer to the desired unit, whatever has been asked in the question. So you have to take care of all these scenarios in numerical problem. For diagram based, you cannot miss on the detail. So for example, they have asked for, let's say N1, N3, and they have asked C4. So you have to see carefully where is C4, you have to see where is N1, you have to see where is N3, you have to observe it properly. For experimental analysis, you have to draw a mental picture. So different ways are there for solving different questions. You cannot just blindly look at the question and try to see from the answers what can be applicable. There is an approach for everything. For arranging, ordering, directly trying to answer from option is not at all recommended. What you need to do is you need to give your own order. Based on what knowledge you have, concept you have, try to get your own order of the questions. Then only you will be able to solve them correctly. Match the following, even if you know 1 or 2, then you will be able to solve them. Graphical questions, I'll give you some tips and tricks what you need to do for graphical questions. And true or false. So true or false, you can use the elimination method that definitely this cannot be correct. or definitely this can be true. So these techniques can be also used. Next tip is selective reading of a question. This I have seen for many students at Biotechnica as well. So our brain tries to perceive what things we know, what we have studied, what part we know. But in a sentence also, you have to read the complete details of the statement, not just some part of the question. Part you should not select, you should see the entire details. Every single word can make a difference. So this point I will tell is the most important point why you get part C questions wrong. I've taken an example over here. This is just one statement from a part C question. Right-handed double helix and anti-conformation. It is to differentiate between A, B, Z forms, A and Z forms over here. So right-handed double helix and anti-conformation for the base to sugar arrangement in aDNA, and left-handed helix with alternating sequence of G and C and alternating syn and anti-configurations for base to sugar arrangement in zDNA. Every detail you have to check. So instead of right-handed, they can give left-handed. Instead of syn and instead of anti, they can give just syn. Instead of G and C, they can give A and T, right? So instead of aDNA, they can make bDNA. You have to be very careful of all these details. There may be another reason why you're getting it wrong is focusing on the speed rather than the accuracy while practicing. When we just get into a habit of we learn any concepts we are learning new, then we try to practice questions right after that. We'll try to see if we are getting it correct or not. But immediately what we will try to do is, immediately you will try to focus on how fast you are able to answer. Okay, as the time goes on, you are going to lose the patience to solve the question. But that shouldn't be a part of it. You have to focus on the accuracy part more. Even if it takes 15 minutes to 20 minutes to solve a part C question, while practicing, during your preparation, you focus on the accuracy. I will say you allow 1 to 2 months until you get all the questions. Most of the questions you will be getting it correct. Wait for the time when you get most of the questions correct. Then only you practice on making it more faster by giving some mock tests. Okay, where you can put a timer and you can measure your accuracy, where you can measure your speed. But first thing first, always focus on the accuracy first, then the speed. Then fourth is wrong selection of questions. So actually question selection is an art. If you ask me, there are 3 types of questions in CSIR. Ones are the direct ones. Direct questions like which of the following is not an extracellular protein? Keratin, laminin, collagen, vitronectin. Or in TCS cycle, the energy rich molecules are what? Like 2 GTP or 1 GTP, 2 NADH or 3 NADH, 1 FADH2 or 2 FADH2. These are all memory based, mostly direct questions. Second will be some tempting type of question. Now tempting is what? You know the answer, but means you know the concept, but you will have a high chance of making a mistake. Those are tempting. And the question setters, they will know also that you have a high chance. Why? It will mostly have a word play in it. Okay. So some activation, inactivation, lot of confusing word plays will be there, because of which you may get it wrong. So you have to be careful for such questions. I am not saying you cannot attend such question, but you have to solve it with utter focus. And the third type of questions are trap questions. Beware of that. Now trap questions, I'll give you an example. Let's say match the following. Generally we expect match the following to have 2 columns, right? But sometimes, and that too, they will have one for this, one for this, one for this. So this is with this is with this, and this is with this. So easy. You will have A for 1 or B for 3 and C for 2. But sometimes what they will give additional columns. Okay. Sometimes what they will additional column, but with only 2 factors. That means this can be with this also, it can be with this also. And that is where complications come. And those are trap questions. If you know the fundamental very nicely, then it is fine, else better to avoid such kind of questions. So what type of questions you might select? So this is the tip, what type of question selection you might look out for. One is the direct knowledge questions, which is based on your memory. Okay. Now that depends on what units you have covered or what topics you have covered, you can pick that up. Try to attend more match the following and more graph based questions, because visually what we see, we have a better chance of solving them, but again stay clear of any confusing or trap questions like this. Or else you can go for easy option combination. Sometimes you will find the true or false statements, you can have the true or false statements as only A, okay, or you will have A, B, C, and then you will have A and B, or you will have only D. So this is confusing, right? This is not such an easy option. Whereas you will have some places A and B, C and D, A and C, and B and D. Now you know any two has to be correct or any two has to be wrong. So if you get two wrong, then definitely you will get the others correctly. But in these cases, you don't know. Either this can be only one statement can be correct, only two can be correct, three can be correct. So this is not such an easy option. So better to select. In part C, you are getting total 75 questions. I think you will get a lot of options under that to select accordingly. So you're going for an easy option questions. And second is short introduction part C questions. Don't go for the ones which have long paragraphs written. Mostly you will have the experimental types with long paragraphs, but sometimes again don't be fooled by the length of the question. Some part C questions can look very long, but while solving you will see that most of it is unnecessary. So you should develop the knack of recognizing those questions immediately, which are given unnecessary information, and the questions which are actually the part C length is longer and all of this information will be required. So the ones which will require this information, you can skip those. And another one is similar sentence questions. Like everything in two statements is same except one word. So maybe absence and presence is only the difference, or present in cytosol or present in cortex. That's the difference. Okay, so this kind of similar sentence questions, it will be easy to point out the difference. The rest of all the statements will be true. These are just some of the tips what you can do. Next reason is overlooking the requirement of the question. This also is a major reason for not able to score. So you actually fail to check whether they are asking for correct statement or they are asking for incorrect statement. Major mistakes, silly mistakes for most of the students. Immediately, for the first thing you should look in the question true or false is whether they are asking for correct or incorrect. You don't check which unit they have asked for numerical question. Probably they will ask for expressing in nanometer, but you have expressed in micrometer. And there will be a value which has come same as what you have got as micrometer. So you will very happily select it. But then there will be also an answer which is expressed in nanometer, and that would be the correct answer. Sometimes you choose combination. So as I said, sometimes they will ask only one statement will be correct. In true or false combination questions, they will say only one statement is correct. So you have to see that it's not all correct, it's not all incorrect, only one statement. That means along with the combination, let's say A, B, D, maybe A is correct, but D and A has to be wrong. Like that. Okay, so very rarely that has come, but we have got such questions. You have to see whether if it is ordering or arranging, you have to see the increasing or decreasing signs in the options. These are all overlooking what is the requirement. For example, if you have to arrange the TM in increasing order. Now, increasing order means what? The highest should be at this side and lowest should be at this side. This is what we know as increasing order. But if you see over here, they have given the sign like this, which means this side is the highest and this is the lowest. But what generally people will tend to do is they will miss out the sign. They will think that, okay, lowest to highest, this should be lowest and this should be highest. So based on that, DOPC is lowest. They may tend to pick this one. But look at the signs also. These are the common places where maximum mistakes will take place. Okay, so this again, I can classify it as a trap or tempting, because this you may know, but you may have a chance of making a mistake. Okay. Next, unable to imagine the scenario, question scenario. So in experimental type of questions, you should be able to draw a mental picture. Like if in a mitochondrial experiment, you have to add some factors like ATP, API or dinitrophenol. So if you have taken a test tube, then what you have added, right? So then you can just draw a mental picture. Okay, then what I have added, is it a separate tube or it is like in the same tube? Then you can put an arrow like this. Or if it is a pathway, then you can draw the pathway that this is activating this, you put an arrow and write that, and that in turn is inhibiting this, so you put an arrow and write that. So these things are going to help you draw a mental picture. People don't do that and directly try to answer which options are correct. So if required, you can draw a rough sketch or a picture in your in your rough diagram, or simply a flow chart you can draw to make the process clear, so that choosing the answer will be easier. Now this applies for analytical type of questions, experimental analysis. And also if you have the mutational phenotype, if this gene is not there, then what will happen? Or transplantation, if you take these cells and put it somewhere else, then what will happen? So I suggest you actually draw the embryo and okay, I have these cells over here, I have put it over here. Then what is going to happen? If you draw a mental picture, then it will be easier not to make mistake. The seventh point is keeping this section to solve at the end. Now see, part C is all about analytical thinking. Your brain needs to be fresh, and that's why it's my personal recommendation that solve this section first. Ideally, you should keep 1.5 hours for all the part C questions, the first 1.5, then the next 1 hour for part B question, and the 20 minutes for part C, and last part A, 20 minutes for part A, and last 10 minutes you can keep for revision checks. Okay, so first 1.5 hour, you have to solve 25 questions, that's all. If you don't finish 25 questions, then you move to part C. Okay, you don't complete part C, then you will miss out on the correct answers of part B and part A, which you could have got it correct. So this is the seventh point again. Try to solve part C when your brain is fresh. And the last point is missing out on details and lack of observation. You have to look at the many details what you have to pick up. For example, look at this chart, right? You have a host region and you have a donor region. So host region is supposed to form prospective neuron. Donor region is supposed to form prospective epidermis. If you take the donor, so you can draw an arrow like this, right? And put it in host. The simple thing will make it easier not to make a mistake. That means what? The epidermis, even with neuron, it is making epidermis, which means it is autonomous specification. But if you just change the headings, so what our brain will tend to analyze, if I just close this, right? And there were some other headings, some students don't even see the heading. Then it will look like prospective neuron taken into prospective epidermis, and it changed the specification to epidermis. Then it would have become conditional. With different heading, it will become different. So some students don't look at the headings of the table. That is wrong. Okay. So let's say there is a statement, bond between the phosphate group and the ribose sugar in a nucleotide is called as what? So if you are not aware, this bond is actually called as phosphoester bond, because in a nucleotide you only have one sugar connected with the phosphate. Yes, in a nucleotide chain, in a DNA chain, nucleic acid chain, the phosphate may be connected with two sugars, and in that case it is called as phosphodiester bond. Got it? But in only the nucleotide it is called as monoester. So these are like attention to details what you are missing out. Okay, you know that it is some ester and you are blindly clicking it. Then second, I told you some points you should know. Like headings of the table, similarly x and y axis of the graph. When you look at a graph based question, immediately you should check what is there in y axis, what is there in x axis. Then you look at the body of the graph, what is the pattern and all. Okay. Similarly, headings of table, pay an attention to that. Another thing is play of words, like whether it is saying activation or inactivation. Like if it says allosteric activator of this enzyme. Now somewhere you have read allosteric is correct. So your brain is selective reading it. That allosteric, okay, it is correct. But then further, whether it is activating or inactivating, is that part correct? Or presence or absence, we tend to miss out on. Obvious details we will tend to miss out on. Right? Lack of observation. Phosphorylation, dephosphorylation. A small word, dephosphorylation, that will be missed out. That can be missed out. So these were the tips from my end. I just want you to remember that get your concepts right and practice till you get perfect, and then I'm sure you will score much more marks in part C. With that, thank you for listening to this video. I hope it was helpful.